When an ant is infected with a pathogen, something very strange happens. The exposed ant is groomed by other ants to help spread the pathogen through other members of the colony. And they do this by licking each other. So why don't we do this? We instead take the exact opposite approach and deliberately avoid licking each other while we are sick. Actually, we do a very similar thing, because essentially what these ants are doing is inoculation, which is a less controlled version of vaccination. It's what we did ourselves before we had more modern technology. Inoculation is exposure to the virus or pathogen in small doses in hopes that this prepares the immune system and wards off the actual disease itself. An example of inoculation is the chickenpox parties we used to have prior to the introduction of the varicella vaccine in 1995. Both methods, inoculation and vaccination, increase the immunity of a population through exposure. Inoculation is just much more risky than vaccination. So let's talk about the vaccines we've been hearing so much about lately. But first, real quick, what's a virus? Viruses are essentially just a piece of genetic material with one primary goal: to reproduce. While some may argue that this applies to every living thing, viruses are a special case because they can't replicate by themselves. Viruses are just pieces of DNA or RNA inside a protein capsule just floating around waiting till it finds a host. Once they're in though, they can wreak havoc. They take over by hijacking the cell's machinery to enable the virus to make more of itself. The host has a backup plan though. Specifically, the immune system notices something's not right and generally reacts like this. So vaccines take advantage of this immune response and use a piece of the virus to prepare for it. After a vaccine, you may spend a few days feeling under the weather, and this is normal and can be a sign that the immune system is hard at work taking note of these foreign proteins, which are also called antigens, for future reference.
How many times do we have to teach you this lesson?
Okay, so now to the coronavirus vaccines. There are 3 types of SARS-CoV-2 vaccines that work by slightly different mechanisms. But what they all have in common is an ability to introduce the body in a controlled way to the spike protein. So why the spike protein? One reason it's such a good target is because this protein is on the surface of the virus, making it one of the first things the body encounters when the virus tries to enter the cell. Okay, now we got that out of the way, let me give you a too long didn't read of the 3 different vaccine technologies. The first one is Johnson & Johnson, which delivers the DNA encoding the spike protein in an inviable flu vaccine shell. Pfizer and Moderna has the mRNA encoding the spike protein. And Novavax contains the straight up spike protein itself. Wow, we got the whole central dogma going on here. Okay, so now let's get into a bit more detail about these vaccine technologies. The first vaccines to get emergency approval in the US are the mRNA vaccines, which include Pfizer and Moderna. mRNA, or messenger RNA, is the instructions the cell uses to build proteins. These mRNA vaccines contain only the instructions needed to build the spike protein and nothing else. And what's amazing is that the machinery our cells uses to make proteins, the ribosome, doesn't care where the mRNA came from, it just does its job and makes the protein. After getting the vaccine, our cells take up these mRNA instructions and build the encoded protein and then present it on our cell's surface. Without the rest of the viral instructions, this protein just kind of sticks out like a sore thumb, which then alerts the immune system that something's not right here, which then the immune system takes note and is prepared for next time. Next, we have the Johnson & Johnson vaccine. This vaccine uses a viral vector to deliver the DNA encoding the SARS-CoV-2 spike protein. A viral vector approach means using an inactivated virus to deliver the genetic material. And in this case, the inactivated virus is an adenovirus, which causes the common cold. This altered virus is harmless because it can't actually replicate in our cells. But the DNA it contains can be converted into mRNA, which can be converted into the spike protein by our cell's machinery. This imposter virus serves to produce the proteins that will allow our cells to recognize the actual coronavirus in the future. And the third type, which is used by Novavax, contains the spike protein, skipping the DNA and mRNA steps completely. This vaccine contains the spike protein itself and works by exposing the immune system to the same protein it would see if the coronavirus invaded. And just like the other two vaccines, any antibodies that are generated by the immune system against this lone protein will also take out the protein when it's attached to the viral surface. No matter what vaccine is used, vaccination teaches the body to recognize a specific intruder more quickly, ensuring that any future infection will be properly taken care of. With adequate vaccination, we can follow these ants' leads towards herd immunity without having to take on some of their weirder habits. And with that, thanks for watching.